Thank you for your kind introduction. Today, I would like to thank organized members for giving me this opportunity. Actually, I am an analytical chemist, so today I will talk about the comprehensive analysis of the microbiome lipidome. And unfortunately, I don't have time to talk about the C13 experiments. But if you are interested in this topic, so please let me know. Okay. As you guys already know that there are many bacteria in our gut, and they regulate our homeostasis and disease. So our global question is, how gut microbiome affect the host phenotype? And one of the possible mechanisms are metabolites and lipids, as the previous speaker also said that. So I will explain one of the brief example. Most famous one is short-chain fatty acid. They affect the GPCR or 43 and 109A, and also butyrate affect to the goblet cell, inhibit the H activity and induce the Treg differentiation. And also there are some reports about the long-chain fatty acid metabolism by microbiota. Recently, the fatty acids are conjugated by amino acid, and they affect act on the GPR119 and increase the glucose homeostasis. And also, the hydroxylated or oxo form of linoleic acid affect to the GPR48 or 120 or trip channel, and they control the energy expenditure or glucose homeostasis. But these are very famous examples. So we need to know the whole view of metabolites in gut microbiome. But knowledge is limited because sequencing has been developing, but the prediction of metabolic pathway is still difficult because there are many hypothetical proteins. So we need to see the molecule directly. So metabolomics and lipidomics is a promising approach. Actually, I'm a lipidomics person because I like lipids, and the reason is lipids are very good classification index because before the sequencing has developed, the chemical component is used for the classification index because the bonding type or fatty acid composition or chain length or kinones are very unique for individual species. So these lipids are good indicator of species. And we higher animals or plants have the specific receptors such as pattern recognition receptors to sense the invasion or infection of such microbes. So these lipids can be a signaling molecule. So I'm working on the lipidomics studies. Actually, this is lipidomics studies use the mass spec in general. In LC, we separate the complex samples, and next the components are ionized, and the mass spec fragmented lipids to small fragment, and we can see this kind of data. This is called MSMS spectra. This spectra shows the molecular structures. Then we need to annotate this and interpret this data. Then we can estimate the structure if we are lucky. Actually, this process is very difficult and time-consuming. So currently, there are two ways of lipidomics. First one is targeted analysis. This means this analysis is for known metabolites only. So this is very easy, high throughput, and high accuracy. But this targeted analysis may miss the unique metabolites. So I'm working on the untargeted way. So today, I will talk about two topics of untargeted analysis. First one is the lipidome. This is a component of LPS in bacteroides strains, cultured bacteria. Then next, I will talk about the lipidome profiling in mice feces. The most famous one of the bioactive lipid in bacteria is LPS. This is the gram-negative bacteria-specific lipid, and the polysaccharide is bind to the bilayer by this anchor molecule called lipidome. This is the structure of E. coli type lipidome. This has the diglucosamine backbone with 2 phosphate groups and 6 acyl chains in total. And this is the very strong agonist of TLR4. So this induces the inflammation. But actually, there are many gram-negative bacteria in our gut. So why inflammation doesn't happen in the stationary condition? Actually, the previous studies show that the bacteroidetes-derived LPS didn't induce the inflammation cytokine expression on PB, peripheral blood monocyte. And also, the bacteroides-derived LPS inhibit the E. coli-derived LPS signal, because the mixing ratio of Bacteroides E. coli LPS ratio decreasing the inflammation cytokine expression. So, and this is explained by the difference of the structure. Actually, E. coli has this 2-diphosphor hexaacylated lipidome, and the chain lengths are almost 14. And on the other hand, Bacteroides has different structure lipidome, 1 phosphate group and 5 acyl chains. This is the example of Bacteroides fragilis. And this structure may inhibit the signals. So, but actually this is one of the example of Bacteroides fragilis, and other cases not examined. So in this study, I tried to resolve the structure of Bacteroides-derived lipidome structures. Firstly, we pick up the 9 representative Bacteroides strains shown here, very common Bacteroides in human gut, and there are related Bacteroides, Parabacteroides, and Prevotera. And we culture these strains in anaerobic chamber and extracted the whole LPS fraction, then removed the polysaccharide part by acid hydrolysis, then we got the crude lipidome. Then we analyzed this crude lipidome, but we developed a special experimental system because this phosphate compound is very easy to stick to the stainless steel piping, and we cannot get sharp peak. So we optimized the solvent conditioning LCMS system. Actually, I examined the basic alkaline condition by adding ammonia in the solvent. Then we can get very sharp peak. Okay. Then we analyzed the cultured bacteria shown here. Actually, previous studies show that the Bacteroides fragilis have the monophospho-pentaacylated lipidome. And we successfully find this one. This is the previously known structure of mass. Then, but surprisingly, there are many other peaks around here, and these have the 14 Dalton mass gap. So this may have the heterogeneity of the acyl chains. So I tried to estimate the structures. This is the most abundant one, this one. We interpret the MSMS spectra like this. Then we estimate the fragment structure like this. Then we successfully estimated that this is have the 1 phosphate and 5 acyl chains, and the chain length is 17 or 16, and secondary position is 15. And also, we look for this one. And this result shows that the chain length is different. Actually, the main chain was 16 or 17. Then this one is maybe 15 or 16. And this is the mixture of these isomers. Then we look for these, we obtain these data. Actually, this is more complex and heterogeneity because the main structure is not this black one, because this 14,000 smaller molecule is the most abundant species, and even brevotocopri has 28,000 smaller one is the main metabolite. So we compare the composition of the lipidome molecules, and we can see here that the composition is clearly correlated with the phylogenetic different classification, because the common bacteroid strains have the this green one is the most abundant species, and but of these other bacteroids has the other type shown, like this 18.0. So this means that the lipidoid is very heterogeneous. I think E. coli has a single lipidoid type in stationary condition, but bacteroidetes has very unique lipidoid features. So this means that we need to see the molecules, not just the species. So this is the case of the cultured strains. So let's move on to the next in vivo studies. So next question is, what structure of lipids are in the intestine? Actually, we can profile the lipids in the feces, but it's complex because fecal lipidomes can be derived from the food, bacteria, and host. So we can profile, but we cannot tell which is the bacterial lipids, because amino acid is amino acid, so we cannot distinguish the origin. So we need to devise the experimental system. So we use the antibiotics treatment. If we find the lipid present in normal water-treated mice, but absent or decreased in ABX antibiotics-treated mice, this decrease can be explained by the elimination of commensals. So we can narrow down the candidate of bacteria-derived lipids. So we treated the mice with 2 weeks of, then homogenize the whole feces, then extract the whole lipid, then analyze in untargeted manner. This is the overview of the result. We found around 6,000 ion features. This is the volcano plot. Right side indicates the metabolites increased in antibiotics, and left one means the decreased metabolites. So many metabolites was decreased in antibiotics treated group. First, we try to identify the molecules using the conventional database. So we successfully annotate only 274 molecules. Actually, the identified metabolites are mainly bile acid, sphingolipid, and phospholipid, and etc. And we find the bile acid here, and also these are all unconjugated bile acid. And this slightly increased metabolite was taurin conjugated bile acid. Actually, this is very reasonable result because the deconjugation and dehydration of this hydrolytic group is metabolized by intestinal bacteria, and by administrating the antibiotics, these reaction can be reduced. So this primary bile acid or secondary bile acid, the unconjugated forms are decreased. Then the conjugated ones are decreased. So this is very reasonable, and this means our experimental system works well. But problem is, we just only identify around 10% of the intestinal metabolite or lipids. So how to increase the annotation is the next question. So actually, as I showed previously in the case of lipid A, we need to see the spectra deeply. Then we need to understand and interpret what this means. So, but actually we need to see more than thousands of spectra, but it's very time consuming and nobody wants to do that. So I use informatics. So actually the spectra, this spectra is derived from the structure. So same or similar structure produce the similar spectra. So I classify the spectra before time-consuming structure estimation. Actually, the similar structure is assembled to the single network, and this different type of spectra means structure is also different. So we can assemble the similar derivatives. Then we can focus on these metabolites. So we apply this feature-based molecular spectral networking method. And this is the whole view of the lipidome. Actually, the symbols are metabolic features, and the upper and lower triangle indicate the increased or decreased metabolites more than 10 holds. And we first apply the conventional annotation data. As you can see here, these conventional phospholipid, PI, PE, PG are assembled to the single cluster. So this means our approach is successful. And also, we find these subclusters. This shows the bacteria-dependent decrease. So these should be analyzed or interpreted. It has a higher priority for structure estimation. Too long time short, actually we can successfully estimate the structures. And these molecules have unique features. For example, these phosphoglycerol, but their linkage to acyl chain is not ester but ether type, and also the sugar conjugated diacyl glycerol shown here, and also ether type and di-sugar type is also found. And also sphingolipid have unique features such as this type has the phosphoethanolamine conjugated sphingolipid and phosphoinositol conjugated lipid, and also hydroxylated ceramide, and also further H-rated, H-rated ceramide is also found. And also the sulfur carbon chain linkage is also found. This is called sulfonlipid, and this can be assumed as the analog of sphingolipid. So this is a very unique feature of the bacterial metabolites. So next, I also found that FAFA. This one has showed the bacterial decrease, but this is not. So we looked into this deeply. FAFA is the abbreviation of the fatty acid ester of hydroxy fatty acid, and this is like the fatty acid linked to the hydroxy fatty acid. Actually, we cannot predict the hydrolysis position correctly, but the combination of the fatty acid is a long-chain one, like this. And this don't show the bacterial dependence. On the other hand, what we found is the FAFA with short-chain fatty acid and long-chain hydrolysis fatty acid, like this. And all this cluster shows this long-chain and short-chain combination. And uniquely, the hydrolysis position is alpha. Actually, we cannot find this molecule in any database or previous studies, so we named this SL-alpha hydrolysis fatty acid as alpha. We need to validate this estimated structure, so we synthesize the chemical compound of alpha, and we analyze this compound, and we can successfully identify the similarity of spectra and retention time. So we can validate the estimated structure of alpha. So this analysis can reveal the potential of finding the new molecules. And actually, mass spec database is currently very poor because the database can be developed for the commercially available standards. So these bacterial-derived lipids are not available commercially. So generally we cannot validate the structure, but we include these MSMS spectral data into the very famous lipidomics software collaboration with Dr. Tsugawa. And now everybody can use this method, and you can identify these molecules. And this process is explained in this paper. Actually, Dr. Tsugawa applied the software for various kind of samples, such as culture cell, organs, and feces sample. And this color map shows the identification numbers of molecules. So yellow one means the abundant one. And this x-axis means the lipid class. So as you can see here, feces profile is totally different from other samples. And these molecules only exist in feces. And these are one of the examples. So we need to profile the lipids not only for these common samples. We need to perform the untargeted way to capture these unique lipids. So let me conclude this presentation. Actually, gut microbiome produces structurally diverse lipids, and untargeted mass spectrometry have a potential of revealing unexpected bacterial molecules. So take-home message is we need to open our eyes to the unknown. So finally, I want to acknowledge my collaborators. Actually, my boss is Fumio Matsuda in Osaka University, and this works collaboration with Makoto Arita in Riken, and the second topic was mainly contributed by Shu Yasuda, and the mass spec was supported by Shimazu Corporation. Thank you. That's all. I'm happy to take any questions.